member who happens to be the Lincoln, this is the GCR, but he's on his way. He said to start without him, so we're gonna start. It's a great pleasure today to introduce Val Sawiccy for her PhD defense talk. It is a good omen today because I would like to report that after trying for probably 4 years with lots of frustration, we had anemone spawning overnight. Good omen. They were supposed to spawn and they did. So that is good news. So anyway, so Val, I think we're done. No. So Val came here from an interesting academic path. She was a genetics major at University of Wisconsin-Madison. He has arrived. And she then did a big adventure and she went to Guam, where she got a master's degree in real coral reef sort of ecology. So she really took a left turn from her genetics, her more traditional sort of cell and molecular genetics path. And from there, she became really interested in combining those two interests. So the more the field-based interest in coral ecosystems with the molecular biology and genetics. And she came to my lab. And she's been here a while, let's just say it that way. But there's some good reasons for that. There's this whole pandemic thing that you've probably heard about. But also, I'm allowed to share that for each year that Val was here, she had an ear surgery. So she's had 7 surgeries during her PhD. Amazing. And yet here we are. She has a great talk to give and a great story to tell about her thesis. And she has overcome. And she is going to be starting an instructional faculty position in January. So chop, chop, Val, at the University of Oregon. Boo. Anatomy and physiology, but we'll still let you come and visit the lab. Anyway, so please, Val, here is your thesis defense talk, metabolic and immune responses in Cnidarian symbiosis and dysbiosis. Val.
Thank you for the introduction. Yeah. Okay, yes, so my thesis is on symbiosis. So I just want to give you a really quick definition in case you don't know what that is, but essentially one definition is that it is the association of 2 or more organisms. So what my thesis is really focused on Cnidarian algal symbiosis. So it involves cnidarians such as corals or sea anemones and their partnership with dinoflagellate algae from the family Symbiodiniaceae. So this forms a persistent partnership that's mutualistic. So they're both benefiting each other. So the cnidarian host shares inorganic carbon with the algal symbionts, and it also provides a residence for these algae that's in a high light environment, and it offers them protection as well. And in return, the algal symbiont will provide the cnidarian hosts with oxygen and photosynthetic products such as glucose. Now, it's this cnidarian algal symbiosis and this mutualistic exchange that is the foundation of coral reefs and the coral reef ecosystems. So corals that you kind of see here form these these like 3D habitat structures, and they're builders. So they form a lot of habitat. And they're dependent on this cnidarian algal symbiosis. And they're critical for cultural reasons, ecological reasons, and economic reasons. So corals are, about 500 million people rely on coral reefs for either income, for a food source, or coastal protection. And their estimated worth is $11 trillion, if you were to put a number on it. And also they're hotspots of biodiversity. They're actually the most biodiverse ecosystem that we have on Earth, and they are home to about 25% of marine life. However, this habitat, these ecosystems are sensitive to environmental shifts, particularly those in elevated temperatures. So during elevated temperatures, there's a breakdown of this Cnidarian algal symbiosis, and this results in a phenomenon known as bleaching. So in this picture on the left here, we see a pigmented coral, and I see that brown pigmentation, that's because of the algae. However, during prolonged heat stress, we see these corals begin to pale because this symbiosis is breaking down and they're losing their algal symbionts. And this white color is actually their skeleton underneath. And because the algal symbionts are their main source, or their main nutritive source of energy, because of they're giving them the photosynthetic products, they actually will start to starve during this heat stress. And if the waters don't cool back down, they actually will often just completely die. So here on the picture on the right is a dead reef. It's just coral skeletons that are covered in macroalgae, and these will eventually turn into rubble fields that essentially removes this critical three-dimensional habitat for marine life. So this is a graph of the temperature anomalies ranging from the years 1880 to 2022. And so, and on the vertical axis here, we have degrees, 1 degree to negative 0.6 degrees. And what we see here starting around 1980 in red here is like an increase in the temperature anomalies that we're seeing. And these correspond to global coral bleaching events where this bleaching phenomenon is happening worldwide around coral reefs. And the first one was started in 1998, and there was one just last year. So during my dissertation, there's actually been 2 global coral bleaching events. And these events are increasing in severity and frequency, and this is causing a coral reef crisis due to the mortality of these critical ecosystems. So thus, it's really important to understand the interactions between cnidarians and the hosts and these algal symbionts, because we're losing this critical habitat. And so there's a need to develop some kind of solutions to mitigate the effects of climate change that are having on corals. So my thesis is understanding this interaction between cnidarian hosts and algal symbionts. So before, I'd like to talk a little bit more about this symbiosis, but to do that, I actually need to introduce some cnidarian anatomy. So cnidarians are composed of 2 tissue layers. The outer layer here is the epidermis. The inner tissue layer is the gastrodermis, and if you notice the brown spots, that's where the algae reside. And these 2 tissue layers form a compartment known as the gastrovascular cavity. And if we were to zoom into these tissue layers, we can see this algal symbiont. It's actually residing inside of a host gastrodermal cell. So it's actually inside of the cell of the host. And it actually resides even more in this membrane-bound structure that we term a symbiosome. And so this is a very intimate association. This is actually known as an endosymbiosis, and it involves cnidarian immunity and metabolism. And that's a very intimate association. So so similar to corals, the sea anemones Exoptasia diaphana form a similar endosymbiotic partnership with these algal symbionts from the family Symbiodiniaceae. So I'll be using this as a model for my thesis when studying the symbiosis. And instead of referring to the species name, there's a common name called Aptasia, and I'll refer to it as Aptasia herein. So one of the reasons I'm using this model is because unlike corals, these cnidarians these sea anemones can be in the state with symbionts or without symbionts, making it like almost a natural negative control. In addition, they can undergo bleaching due to this prolonged heat stress where they actually lose their algal symbionts, similar to coral. Okay. Now, there's 2 different conditions that my thesis is going to be looking into that I want to kind of define. The first one is symbiotic state. This is essentially, is the anemone symbiotic or not? So symbiotic anemones are those that have the algal symbionts, and aposymbiotic anemones do not have algal symbionts present. The other term or condition that I'm examining is dysbiosis, and this is essentially symbiosis dysfunction. And I'm going to be looking at dysbiosis as a result of induced by heat stress. Okay, so now I want to kind of look a little bit more about what's happening during these 2 different conditions. So during symbiosis, we have a host gastrodermal cell here pictured that is symbiotic with the algae during ambient conditions. And we see that the host is sharing inorganic carbon as well as phosphorus and nitrogen, these nutrients with the algal symbiont, and then the algal symbiont is sharing sugars with the host. However, during dysbiosis induced by heat stress, what we see is there is a reduced translocation of this sugar to the host. So the host begins to starve. And this becomes, can become prolonged and actually cause a result of the loss of algae and starvation and death of the cnidarian host. Now let's look at immunity a little bit closer. So what's happening at these same conditions, but with respect to the immune response of the host. So again, here's a host gastrodermal cell that is symbiotic under ambient conditions. And what we see is that there is a downregulation of these or suppressed immune host responses, specifically of autophagy, apoptosis, and phagosomal maturation. So it's that when the algal symbionts are present, they're somehow modulating the host immunity, suppressing these immune responses. However, during dysbiosis, what we see is that these processes are actually re-engaged. And during subsequent heat stress and during this algal loss, it's possible that these host immune responses are contributing to the loss of algae, but that remains to be, that's poorly stood right now. Okay, so my dissertation is going to occur in 2 vignettes or 2 parts. So I'm going to be looking at these 2 conditions during immunity, or looking at immune response of the host and also the metabolic response of the host. So I'm first going to start with immunity in chapter 1, and then I'll go into metabolism later. Okay, so I'm going to be investigating an immune gene known as NADPH oxidase. Also termed as just NOX, which is how I'll refer to it. But why do I care about NOX? Well, NOX was identified as a candidate symbiosis gene, and it's found to be upregulated in bleaching corals and sea anemones. And also we found that a NOX gene was upregulated in the gastrodermis tissue that contains the algal symbionts relative to that outer epidermal layer. So this suggests that somehow NOX is related to symbiosis. So what we know about NOX thus far is mostly found in vertebrates, and NOX functions as a stress response and an immune defense against like foreign invaders such as pathogenic bacteria, for example, in phagocytes or some kind of vertebrate immune cell. And it's involved in the phagosomal maturation process, which is what you see pictured here. So in purple is some kind of foreign invader, like a microbe. And during this maturation process, we see that the microbe is located inside the phagosome. And then this phagosome then kind of combines with the lysosome to form a phagolysosome. So this is that maturation process. And it's known that NOX is kind of a marker of this late phagosomal maturation process in the phagolysosome specifically. And they also know that it participates in actually degrading these foreign invaders by producing reactive oxygen species that I'll refer to as ROS. Now, the NOX gene is actually composed of many subunits. It's not just one protein, it's actually several protein subunits that form a NOX complex. So this is just a canonical NOX protein subunits in vertebrates that I'm just going to introduce you to. So they have 2 membrane-bound subunits, NOX2 here in yellow and P22-FOX in green. And then in the cytoplasm are P67-FOX and P47-FOX in blue here. And these are just kind of dispersed in the cytoplasm when it's inactive, meaning there's no immune response happening. And then when there is some kind of response that activates them, these activator proteins will translocate from the cytoplasm and dock onto these membrane proteins. And then this will become an active complex that will produce superoxide by this catalytic subunit NOX2, and that will form downstream reactive oxygen species that will again degrade some kind of foreign invader. So based on what we know of vertebrates and the previous studies that found that NOX was somehow involved in symbiosis, it's natural to ask what is NOX's role in symbiotic cnidarians. So I hypothesized that NOX plays a role in the regulation of the cnidarian algal symbiosis, and it's somehow involved in the phagosomal maturation process. So to answer these questions, I approached this using the central dogma of biology. So essentially what it is DNA in the genome codes for some kind of genes, and these then are transcribed into RNA messages, and these messages are then translated into some kind of functional protein. And I decided to investigate all different aspects of this dogma. So the first thing I did was search for NOx genes in the adaptation genome, using the human genes as a reference. And then I quantified the NOx gene expression using reverse transcriptase quantitative qPCR or qPCR. And then I looked at the localization of this protein NOx in the anemones. I also looked at the expression of these NOx proteins, and I looked at the activity of NOx. Now, I want to talk about this in a little bit more detail now. In order to examine the localization, I had to create custom polyclonal antibodies, and I did this for 2 of those subunits, NOx2 and P67 Fox here in yellow and purple. And these were again used to determine to figure out where these antibody targets may be located in the sea anemones. I also used these antibodies to quantify the NOx expression using Western blots. And I also examined NOx activity by quantifying the amount of activity of reactive oxygen species using a label that can actually label this ROS. And I did this in the presence with and without an inhibitor of NOx. Okay, and I examined these questions were, again, I was examining these 2 different conditions. So I looked at symbiotic state for RNA and protein expression. I also looked at heat stress, which induced some kind of dysbiosis response. And I subjected the sea anemones to 5 days of heat stress, and I sampled throughout this timeline. So let's first look at what I found in the genome. So I found 2 different membrane-bound NOx subunits. So NOx2, which is the catalytic subunit, P22 Fox as the other membrane-bound component. And for NOx2, I actually found 3 gene copies, and I just arbitrarily named them 1, 2, and 3 for ease. And I also found the cytosolic activator protein P67 Fox. I did not find P47 Fox in the genome. Okay, so now let's look at the RNA and protein expression of these NOx genes with respect to symbiotic state. So here is a graph looking at the gene expression of all 5 NOx genes of symbiotic anemones relative to aposymbiotic anemones. So the gene expression of aposymbiotic anemones was set to 0 here, and this vertical axis represents relative expression. We see here that NOx2 copy 1 and NOx2 were uninfluenced by symbiosis. However, we see that these NOx2, 3, copy 3, P22 Fox, and P67 Fox were all downregulated in symbiotic anemones relative to aposymbiotic anemones. So this suggests that the presence of the allyl symbionts is somehow modulating the gene expression of these NOx genes. And if we look at the protein expression using these 2 antibodies, we see a similar pattern. So looking at the target of anti-NOx2 copy 3 and anti-P67 Fox, we see a similar trend where the symbiotic anemones had a lower protein expression relative to their aposymbiotics. This is the similar pattern that we saw in the gene expression. Okay, so now let's look at the gene and protein expression of anemones that are undergoing dysbiosis induced by heat stress. So here, each graph here represents the 5 different NOx genes, and I'm looking at gene expression of the heat stress samples relative to the ambient temperature samples. So ambient temperatures were just set to 1 here. So the NOx2 copy 1 and NOx2 were uninfluenced, or there was no significant difference relative to heat stress relative to ambient temperatures. And similarly, we saw this for P67 Fox. Initially, I was kind of surprised because the previous slide showed that P67 was influenced by symbiosis, but it turns out that this gene is constitutively expressed in vertebrates, so it might be the similar case here. Also, NOx2-3 and P22 Fox were significantly different relative to ambient temperatures during days of heat stress 3 and 5. So it just suggests that somehow these genes are being upregulated during heat stress. Now let's look at the protein expression of during heat stress as well. So there was no significant difference in heat stress protein expression or target of anti-P67 Fox relative to ambient temperatures for P67, but we did see a significant difference at day 3 relative to ambient temperatures. So this agrees with the gene expression data that we just looked at. Right, so now I wanted to look at the localization of these antibody targets in the sea anemones. So these are whole anemones that are mounted on the slide. And it occurs during day 0 or ambient temperatures and days 1, 3, and 5 of heat stress. So on the top row here, we have the target of anti-P67 Fox, and the bottom row here, we have anti-NOx2. And during ambient temperatures, we kind of see this diffuse staining pattern of the epidermis and the gastrodermis. And this darker area in the middle here is that gastrovascular cavity I mentioned earlier. And then during the subsequent days of heat stress or prolonged heat stress, what we notice is this localization of this antibody tends to go near the algae. And this is particularly pronounced here at day 5, where we see this intense signal near the algae. And we see something similar for anti-NOx2/3. We kind of see this diffuse staining pattern, and then we start to see increased intensity of these antibody labels during subsequent days of heat stress. So I wanted to get a better look at where these antibodies were actually localized, because this is kind of zoomed out at the tentacle level. I wanted to zoom in. So I did cryo sectioning. So essentially I took thin slices of sea anemones and stained them. And what you're looking at is a sample from day 3 of heat stress. And these are all different. These are the same image, just kind of highlighting different features. So this first column here is the algal autofluorescence. So this is just the algae and their natural autofluorescence that they produce. And what we noted, we can see in white here, these circle shapes, those are the algae. And this top row here is for the anti-NOx2, and this bottom row here is for anti-P67 Fox. So let's now look at the custom antibody. What we notice is that there's a staining pattern that is like in a ring, like around where the algae are. And we see this for both P67 Fox and the target of anti-NOx2 too. And so I have putatively identified this as labeling the symbiosome, such as a distinct ring. And this cyan color here is just the nuclei that's being stained, the DNA that's being stained. And then lastly, this is just a composite image of all of these different labels. Now, I want to draw your attention to the yellow arrows, specifically for the algal autofluorescence and the custom antibody. What I notice is where I see these distinct rings is where there's also kind of like a decreased pigmentation of the algae. So this suggests that the algae may be degraded here because they have less of that natural autofluorescence. Now, this also suggests, thinking about the symbiosome, the phagosomal maturation process, that suggests that these two proteins are involved in that phagosomal maturation process during heat stress. And that somehow these are being regulated at an individual cell level, since we're seeing this response through individual cells. Okay, the last thing I looked at was NOx activity. So I used, again, a label known as cell rocks to label the reactive oxygen species here. And so I have ambient or anemones at ambient temperature in gray and heat stress anemones here in magenta. And I use a negative antioxidant control. So essentially that's sequestering all the reactive oxygen species. And then I have a positive control. And then I have samples of anemones that were exposed to inhibitor and then not exposed to inhibitor. And we see an increase of the reactive oxygen species in both the inhibitor and without the inhibitor. And we do see this like about 50% decrease when we use the inhibitor. So this suggests that about 50% of that, or this much reactive oxygen species, is being produced by the NOx protein. Okay, so let's that was a lot of results. Let's just sum it up a little bit. So when I looked at aposymbiotic anemones and relative to symbiotic, what we saw was that there was a decreased NOx expression when the algae were present. So we kind of see a decreased gene and protein expression. And during dysbiosis induced by heat stress, what we saw was there was an increase in NOx activity as measured through the reactive oxygen species. We saw an increase in the NOx protein and gene expression, and that there was a localization around this putatively identified symbiosome. So altogether to suggest that during dysbiosis, the phagosomal maturation process is engaged, considering that this is a late marker in vertebrates of phagolysosomes. And also that this is being regulated on this individual cell level here. Also, this work is important for understanding how NOx gene evolution and also NOx function in early diverging animals. There's actually not a lot of work done. This might be the first study done on what NOx does, how it functions in early animals. Okay, so now we're going to move on to my second vignette, or my second part of my dissertation, looking at cnidarian metabolism. First, I'm going to start with chapter 2. So all organisms emit some kind of chemical footprint of metabolites, and this is influenced by the changes in the environment. Some of these metabolites are volatile, meaning that they can transfer across from the liquid to the atmosphere, and these are going to be called biogenic volatile organic compounds. I'm just going to refer to them as BVOCs. And collectively, all the BVOCs that are produced by an organism are called the volatile. Now, BVOCs function as metabolic intermediates, meaning they can change into some other different metabolite within a cell. They also are metabolic end products, meaning that they can accumulate in cells. These BVOCs can also cross cell membranes into a different environment. And they also function as signaling molecules. For example, they may serve as some kind of hormone signal. And of the BVOCs that are characterized in marine systems, we see that they influence biogeochemical dynamics, especially those of like carbon or sulfur cycling between the atmosphere and the ocean. And they also influence physiological and ecological processes. So, for instance, some BVOCs may serve as like some kind of cryoprotectant to protect them during heat stress. And they also influence species interactions. So think about chemoattractants or being able to smell your prey. So they do influence ecological processes that way too. Now, volatile-omics is a relatively new field, and this is just in the last 2 years, there's been only 3 papers on symbiotic cnidarians. So this is a very new and evolving field. And considering that cnidarian algal symbiosis relies on that metabolic exchange, I wanted to investigate the volatile of the Aptasia model system. So I used the aposymbiotic anemones and the symbiotic anemones and conducted the volatile of those 2 samples, and also their symbiotic algae species. And these were algal cultures that were azenic, meaning that they're sterile. And I hypothesized that the identities of the BVOCs that are originating from these samples will be different because they'll be reflecting the different metabolic processes that are happening, and even what might be happening that's associated with symbiosis. So how did I do this? So I used a relatively new technique known as proton transfer reaction mass spectrometry. And how it works is you take some kind of liquid sample in a stripping container, and synthetic air is then bubbled to actually bubble out those the BVOCs from the liquid, and then those are directed into the PTR mass spec. And so for my samples, I took the liquid from the anemones, so like anemones that was sitting in seawater for 24 hours. I took those and I poured them into the stripping vessel or the entire algal culture into the stripping vessel. And so when these samples or these BVOCs are pulled, are bubbled out and go into the PTR mass spec, they are reported as BVOCs plus a hydrogen or a proton. So it's a soft ionization technique that just adds a proton to a BVOC, and it's reported as MZ+1 or mass charge ratio +1, which is the proton. And I'm just going to, for ease, refer to these as just BVOCs. Okay, so this technique it will provide us just a snapshot in real time or a comprehensive scan of all the BVOCs that are present. And relative to other mass spec techniques, This is a soft ionization, so it doesn't really fragment a lot of the BVOCs, and you can detect BVOCs at very low abundances. And this is non-invasive. We don't have to actually destroy our samples to look at the volatile ohm. And the data that we get is semi-quantitative, meaning it can be used relatively, so some kind of state relative to another. So in order to process the data that I'm getting out of this machine, the first thing I did was identify all the different peaks that I'm getting in this snapshot of real time. And then I assigned putative identities using a couple different online databases of known MZ or BVOCs. And then I quantified the mean peak abundances. Now, each VOC, I get a graph like this where I'm getting the concentration over time. I'm actually getting that snapshot for 60 cycles, which is equivalent to about 5 minutes. And we can see that the concentration varies a little bit. So the first 4 cycles were tossed out because these are likely contaminated with lab air in the tubing of the mass spec. And then I binned these values and took the highest mean. So in this example, I took the mean peak abundance for bin number 3. So if I were to just look at my data broadly, just to see the differences and the similarities of my different sample types, here in blue, we have my algae, in orangey yellow, we have symbiotic anemones, and in gray, we have aposymbiotic anemones. These are all 3 stand for different replicates, and the circles that are closer together mean that they're more similar, and the circles that are further apart mean that they're more dissimilar. When I ran up Permanova, there was a distinct volatile ohm between the algal cultures and the symbiotic and aposymbiotic anemones. So in total, I found among all the samples 70 BVOCs. And if I were kind of to categorize them by the different sample type, the aposymbiotic anemones, symbiotic, and algae, I found that the symbiotic anemones produce the most unique BVOCs. This might reflect all the different metabolic processes that are associated with symbiosis. And if I categorize them based on what was produced by each individual sample type, and also what perhaps BVOCs are shared between different sample types, I found that there were 30 BVOCs just specifically associated with symbiosis. So these are all great candidates to investigate the metabolic interplay that's happening between the algae and the host. Between the aposymbiotic and the symbiotic anemones, I found 9 BVOCs. These are likely solely produced by the cnidarian host. And then the symbiotic anemones and the algae produce 6 BVOCs. It's likely that these are produced by the algae, or that's where these BVOCs are originating. And instead of just looking at the identities and figuring out what groups they belong to, I also wanted to look at their abundances. So for the BVOCs that are shared between aposymbiotic and symbiotic anemones, there were 16. I just wanted to know if the abundances were different, and only 1 was differentially abundant with respect to symbiotic state. So that kind of tells me that these BVOCs may be correlated to some kind of essential metabolic processes that are happening, which is why we're not seeing a lot of change. Okay, in summary, it should say chapter 2, sorry. I identified 30 candidate BVOCs that are useful for explaining or understanding the metabolic processes that are underlying symbiosis. We found that symbiotic state only minimally influenced the BVOC abundances. And this is the first study to date that examined BVOCs in the azenic algal culture, specifically from the family Symbiodiniaceae. Most studies have like some kind of microbiome in their cultures. Okay, so now let's move on to my last chapter, chapter 3, looking at, again, both symbiotic state and dysbiosis. So considering that we know that cnidarian algal symbiosis is influenced by heat stress and that contributes to algal loss, I wanted to examine the volatile ohm during heat stress. So I was hypothesizing that the diversity of the BVOCs that are present, as well as their abundances, will decrease in response to heat stress. And this is indicative of dysbiosis as well as just metabolic collapse of the host because of this dysbiosis. So in terms of my experimental design, I took aposymbiotic and symbiotic anemones, and I subjected them to days of heat stress, 5 days of heat stress. And I did the same methods that I talked to you about in chapter 2. And I took 24-hour old seawater that was exposed to the anemones for 24 hours, and I put that into the machine and used those as samples. So if again, this is a principal component analysis, again, looking at like the spatial relationship of the data. In triangles, we have symbiotic anemones. In circles, we have aposymbiotic anemones, and the different colors reflect the different days of heat stress, where the more lighter colors are reflecting more subsequent days of heat stress. And if I put confidence intervals, 95% confidence interval around these two different groups, including symbiotic and aposymbiotic anemones, we see that there's a lot of overlap between both of these sample types. So essentially there's a lot of shared, they're volatile, very much similar between the two. And if I group them by days of heat stress and ambient temperatures, while there's some overlap, we still see some kind of distinct groups, specifically for the ambient temperature, showing that essentially volatile is influenced by heat stress. So again, I wanted to kind of categorize some of these BVOCs. So we're looking at, I'm grouping them between ambient temperatures and heat stress. So here's aposymbiotic and symbiotic anemones at ambient temperatures. And then we have aposymbiotic and symbiotic anemones during heat stress. And what we notice is that there's a lot more BVOCs that are produced during heat stress. So the BVOC's richness is highest during heat stress. This could be due to during elevated temperatures, we see an increase in cellular respiration and metabolism, and this could explain why we're seeing more BVOCs here. And I'm going to highlight one group here. So shared between both aposymbiotic and symbiotic anemones during heat stress, we see that there's 55 unique or BVOCs that are exclusive to those categories. And these are all great candidates to investigate for further studies for biomarkers of heat stress. Okay, so moving on from the BVOC identities, I also wanted to look at the BVOC abundances over time. So I conducted a linear effects mix model. So for my response variable, I did the counts per second, or essentially the concentration. And then for my factors or my explanatory variables, I used the sample type, so symbiotic and aposymbiotic anemones. And then also time, which refers to days of heat stress. And then for my random effects, I use essentially the replicate or the tub of the anemones that were used. And in order to be included in this model, the BVOCs had to be present in all sample types, and had to be also present in 2 out of the 3 replicates in order to be included. I also did try doing a model with an interaction effect, but when I ran in ANOVA, there was actually no difference between every single BVOC, so I just went with the simpler model without the interaction effect. So let's look at the results for type. So there were no BVOCs that changed significantly in their overall abundance as a function of symbiotic state. This is a surprising result considering what we know about cnidarian algal symbiosis. So again, it could be that these BVOCs are involved, or some of them are involved in kind of these essential metabolic processes. And we look at time, we saw that there was a change in the overall abundance of 19 BVOCs. And again, this is corresponding to dysbiosis as well as heat or leaching or algal loss that's happening. So of these 19 BVOCs, most of them decreased in their overall abundance. And that suggests to me that this is indicative of some kind of metabolic collapse that's happening. And for the ones that increase, these are really useful biomarkers of heat stress. So a summary of my findings for chapter 3. So we saw that volatile richness increases during heat stress, and we saw that symbiotic state did not influence the overall BVOC abundance. And many BVOCs did decrease in their overall abundance during this heat stress treatment, and some did increase, but these ones are specifically helpful for looking at potential biomarkers of heat stress. So there were some limitations of using this technique. So I cannot confidently assign the BVOCs an identity. I can only give putative identities, and about 25% of the BVOCs detected were unknown. And in addition, many BVOCs that actually share the same mass. So I can't say which is this one or this one. So there's targeted metabolomics that are going to be needed in order to identify and standards in order to investigate those BVOCs more. In addition, while I use azenic algal cultures, the anemones are actually contain a microbiome. I cannot grow sterile sea anemones. So it's difficult to say that the BVOCs are coming from the anemone versus the microbiome. So there's going to be future work that's needed to kind of untangle what microbes are producing these BVOCs or if it's the anemone. And lastly, this is volatile omics is an emerging field, and there's not a lot of well-established bioinformatic resources compared to other omics. So, for example, the biochemical pathways and the interpretation or the biological processes that are associated with these BVOCs, a lot of it is unknown and uncategorized. So I can't really say because there's this BVOC that there's this kind of metabolic process happening. Nonetheless, this is a baseline for targeted volatile omics exploring both BVOC function and biomarker development and how that relates to symbiosis and dysbiosis. And because this is the first study in symbiotic cnidarians using this technique, this PTR mass spec, this is hopefully useful to improve further experimental designs for future studies. And altogether, this improves our understanding of the metabolic and the signaling pathways that might be associated with symbiosis and dysbiosis. Okay, let's summarize my whole thesis in one slide. Okay, so here are my two different conditions. We have symbiotic state and dysbiosis. And let's look at immunity. And when anemones were symbiotic, we saw that there was a decrease in this host immunity as measured by this decreased NOx expression that we detected. And however, during heat stress induced dysbiosis, we saw an increase in the re-engagement of this host immune process as detected by NOx. And we see that there's this immune reaction might be regulated at an individual cell level, and it's engaging the phagosomal maturation process. And in the symbiotic state during when we look at metabolism or the volatile specifically, we saw that there was an increase in the richness of BVOCs. We also saw that there was an increase in BVOC richness during heat-induced dysbiosis. However, in the symbiotic state, there's essentially no change in the BVOC abundances. However, during heat stress-induced dysbiosis, we did see that there were changes in the overall abundance. Most of these BVOCs actually decreased. So what does this all mean? What can I do with this work? So essentially, I'm trying to categorize the metabolic and immune reactions that are happening between cnidarian and algal symbiosis and figure out what's regulating these dynamics. And this is essentially contributing to a body of knowledge of cell bio and what we know between these metabolic and immune reactions between cnidarian and algae. And the idea or the purpose of this work is to understand basic coral biology so we can start to develop tools to help mitigate the effects of climate change on corals. So we can help develop some kind of monitoring or coral reef conservation efforts. Okay. Moving on to my acknowledgements. So first, I just want to thank my collaborators for my cnidarian immunity work, Angela Poole, Lily, and Noah. Lily and Noah were both undergrads that worked with me, and they contributed significantly to this NOx work, and they're both going to be co-authors, which I'm pretty excited about. And I couldn't have done this without them. They've been excellent. Looking at cnidarian metabolism, I collaborated with the Halsey lab and Vesh. You helped me a lot with the data processing and experimental design when working with this new technique. Also, the Phillips lab for helping me with data processing, as well as Sam Bedgood from the Weiss lab. Okay. I would like to thank my advisor, Virginia. There's a lot I want to say. I don't have time to say it. So thank you for keeping the lab super well-funded. I feel like I've been very fortunate to be in a well-funded lab. You have been an inspiration in terms of coral reef research, specifically cell biology, because you know, like I said earlier, there's been 2 coral bleaching events that have happened, and it's been kind of hard to process what is the meaning of my work if there's this ongoing coral reef crisis. And I think you've really put the hope to see, look on the bright side of things, and that's been particularly hard. This other one's kind of more of an anecdote, but I know I came to the lab and I was like, Virginia, I'm so sick of these lists of genes and these transcriptomics. I just want to work on we need to know what these genes are doing. And what's funny is I also ended up creating a list of metabolites. So thank you for forcing me to understand and appreciate the lists and what they may be. And I think the biggest thing I've taken away from you is your ability to storytell. That has helped me teach better, communicate better, and I thank you for sharing that knowledge with me. Okay, I want to thank my committee. They've seen this project evolve over a couple of years, and I think a couple of different versions, but I think everyone gave me sometimes tough feedback, but it helped me create better science and also become a better scientist myself. And particularly, I know you all got me to think more critically about my work, even though it made it more complicated, and also troubleshooting as well. I know I had y'all were super helpful with that. A huge thanks to the teachers we have in this department, our instructors. We have an awesome teaching faculty staff here. And I think I have learned a lot about experiential learning, and they all have unique teaching philosophies that were there I got to learn from, and they've obviously inspired me to go into teaching and later. So thank you all. Huge thanks to the Weiss Lab. You have been there to argue about corals, to cry with me about corals, to help me with lab work questions. Anyways, also just sometimes hang out and play mahjong. So thank you for the emotional support too. I do want to give a special shout out to Shumpei. He has been my office mate for like 5 years, and he is a sharer and keeper of lab specific knowledge, and I would not be here without him. And also there's been a lot of people that kind of have come into the lab, ad hoc lab members and visiting researchers that have helped me too, as well as a whole slew of undergraduate researchers that have contributed to this work. And also none of this would have been possible without our animal care staff. I think they don't often get to see the fruits of their labor, but they're essentially the ground in order to do this work. Lastly, is the office staff. So they have been there when I needed to call EHS to also call facilities. I just am flooding the floor quarterly. So they have been there when I needed them to. And I know there's also a lot of behind the scenes work in terms of getting my thesis together, and also things like GTA assignments that they're doing. And anyways, I appreciate them for helping me through this program. And this is more of kind of a plug, and I also don't think they get thanked a lot, but OSU has so many resources available to us. We are so lucky, and I have used a lot of them, and a lot of them have given me a lot of professional development skills or emotional support sometimes when I need it. So this is more of a plug to all the students that are out there. Please use our resources, they're great. And yes, it does take a community or a village to get a PhD, and I do think they have also contributed. Okay, questions?
Yeah, okay. So open for questions except the committee. We'll ask plenty of trailer. So questions for Control.
Yeah. So the first chapter, you didn't find, was it P47?
P47
right. So what's the distribution of those genes in other genomes? Like, have they been mapped across animal phylogeny?
Yeah, so just recently there's been a couple papers where it's the first time Knox actually shows up in cnidarians. For a long time it was believed that they didn't exist. And anyways, those genes also found those 3 proteins, but P47 doesn't show up. Oh, geez, I don't remember when.
But it's within bilateria that it's
yep. And because there were multiple gene copies, that's actually not uncommon for cnidarians in general. But also there were the other studies also found 3 copies of that gene as well. Yeah.
Great talk. I wanted to ask that, did you find any biomarkers that were associated with heat stress or VOCs in your data?
Yes, so I guess I can't really say that they're a great biomarker until there's some kind of like these post studies, and also we need to verify the identities, obviously. But in particular, in 121, I think there was a slow linear increase over time, and that was just, I think, out of all the BVOCs, that there was like 5 that increased. You remember this pie chart? Hold on, let me go back. There were 5 that increased over time, but one was, they mostly started increasing after 24 to 48 hours. But there was one that just had this like nice linear trend. It was 121 S or MC plus 1 value, and it was had multiple identities, so I can't tell you what it is.
Questions from online? The last skewer valve. Okay, great. Well, good job, Val. Congratulations. Thank you for coming back. The committee can follow us. We'll probably reconvene in 5 minutes, and there will be some snack.